The human body is a vast, highly active ecosystem. We host trillions of unseen microbes, bacteria, archaea, and fungi, all governed by ancient, complex genetic codes that dictate our biology. For decades, identifying these organisms meant trying to grow them in a lab. But this culture-based approach had a massive blind spot. The vast majority of human microbes simply will not grow in a petri dish. If researchers couldn't culture the organism, it remained completely invisible. Researchers turned to targeted DNA methods, like 16S amplicon sequencing, to profile communities without culturing them. But 16S acts like a low-resolution radar. It identifies bacteria down to the genus level, but then drops out. It misses species-level detail, functional genes, and viral data. To capture the missing details, researchers use whole genome shotgun sequencing. By reading all the DNA present in a sample simultaneously, this method achieves the precision required for species-level resolution. Mapping these complete functional blueprints provides the biological context required to link specific bacterial strains directly to their metabolic outputs. Traditional shotgun sequencing requires shattering DNA into tiny, fragmented pieces. Third-generation nanopore sequencing solves this fragmentation problem by processing massive, intact strands of DNA directly. This technology also reduces the required physical footprint. Instead of relying on a room full of massive servers and laser optics, an entire genomic sequencing facility now fits into a modular device the size of a candy bar. Inside that device is a synthetic, electrically resistant membrane. Embedded directly into this membrane are specialized protein nanopores, tiny biological holes that form the physical core of the sequencing process. To sequence a sample, the long DNA strands are first prepared with a single-stranded leader at one end. They are then mixed with a specialized motor enzyme that binds to the sequence and guides it toward the membrane. Older cyclical sequencing chemistries yield short fragments of a fixed length. This system relies on a continuous physical ratcheting mechanism. It pulls intact DNA molecules hundreds of kilobases long straight through the pore, maintaining sequence accuracy from the first base to the last. The innovation of the nanopore is its translation mechanism. It converts a physical, biological molecule directly into a digital electrical signal. The enzyme unzips DNA, feeding a single strand through the nanopore. An applied potential forces ionic current through this tiny aperture. As unique nucleotides pass through, their physical shapes disrupt the flow, creating distinct step changes in the electrical readout graph. By measuring these minute electrical squiggles, we effectively allow electricity itself to feel the shape of genetic letters. Raw physical biology is digitized into actionable data. A single human gut sample produces terabytes of noisy electrical data, representing thousands of overlapping microbial species. Sorting this out requires immense computational power. To simplify and untangle these massive biological graphs, researchers rely on advanced neural net base callers and next-generation algorithmic assemblers, like MiloAsym. These tools process the raw signal and reconstruct the individual genomes hidden inside the sample. Because these algorithms operate on extremely long continuous reads, they can cleanly bridge complex genetic sequences. This allows them to map low-abundance species and differentiate highly similar bacterial strains that short-read platforms would inevitably scramble together. The nanopore streams this data instantly. Instead of waiting days for a fixed run to finish, researchers monitor the assembly in real time. They can halt the machine the moment a specific target, like an antibiotic resistance gene, is confidently identified. Fusing this biophysics with advanced AI allows us to construct metagenome-assembled genomes, or MAGs. These complete, high-resolution master maps give us the exact functional profile of a microbiome. These master maps provide the technical baseline for tracking how individual microbes respond to clinical changes over time.